Hello everyone, this is your instructor Kwame Shen. This lecture is part 3 of the classification of prokaryotes, and we'll cover gram-positive bacteria that have high GC ratio, and also look at gram-negative proteobacteria of the classes alpha proteobacteria and beta proteobacteria. So for the high GC ratio, we have organisms that belong to the phylum Actinobacterium. And this phylum, you have the genuses Corinobacterium and Mycobacterium. And then the group Actinomyces. And Actinomyces, we have the classes Actinomyces, Nocardia, and Streptomyces. So beginning with Corinobacterium, Corinobacterium are pleomorphic, meaning just like the Mycoplasma, they have different shapes, and so they assume different shapes in terms of their morphology. They are facultative anaerobes, or they may be aerobes. They exhibit what we call snapping division, or palisades. And in this picture here, these are palisades bacteria, or Corinobacteria. You'll see them lining side by side. Palisade is used to describe bacteria that will lie side by side, like a fence, like it's a fencing terminology that are used to describe bacteria as well. And they may also have these club edges, assume a V shape as well. They have phosphate inclusions, so metachromatic granules, which serves as reservoir of energy and phosphates. And an important representative of this group is a Corinobacteria diphtheria. Corinobacteria diphtheria is a common infection in children. It causes infection in children, and so we have a vaccine, the DTaP, the diphtheria, tetanus, and acellular pertussis vaccine, which all infants are immunized against to serve as an important protection for that group. It is also characterized by grayish membrane in the throat. So as you can see in this picture, you see this membrane that is formed in the throat of this individual. And it is very characteristic of Corinobacterium diphtheria. It is made up of fibrin. Fibrin is found in the blood, a protein that is part of the clotting agents in the blood, dead tissue, and bacterial cells that will come together to form this. And they can actually end up blocking the air passages to the lungs. Then another important group would be the Mycobacterium. The genus Mycobacterium are rod-shaped. They may be curved, but they are aerobic. They are characterized by slow growth. So they grow in a very slow fashion. They have cell walls that are made up of mycolic acid. So they have cell membrane, peptidoglycan, and some other components of carbohydrates, galactican or galactan. And then, but more importantly, they have mycolic acids. Mycolic acids gives them a lot of resistance. It's waxy, it's made up of lipids, and so it gives them that protection. And so when it comes to staining Mycobacterium, we use acid-fast staining, where we may use either heat or chemical means to try to infuse the dye into the cell membrane. Important representatives: Mycobacterium tuberculosis, Mycobacterium leprae. Mycobacterium leprae, up to now, we are not able to culture it. Mycobacterium tuberculosis was identified in 1882 by Robert Koch. So that was a very long time ago. Mycobacterium tuberculosis is implicated in tuberculosis, which is a lung disease. It could also affect other parts of the body apart from the lungs, but it could lead to scarring of the lung tissues, as you can see in this picture here. For Mycobacterium leprae could lead to the loss of the digits. So there is a serious loss of tissue in terms of the hands and sometimes the foot as well. Then you have a group of organisms as part of the gram-positive high GC contents known as the Actinomyces. They are filamentous. They form spores, but their spores are not for reproductive reasons. It's mainly for survival. And within this group, we have the classes Actinomyces, which is a non-spore forming group within this group, but they are facultative anaerobes. So they will form these hyphae-like edges or ends, just like fungi, And then you have Streptococcus, which is an important bacteria that is used in the production of important pharmaceutical agents, latex and pesticides and the likes. And they are also used in producing antibiotics. Then you have Nocardia. Nocardia is mainly found in the soil and water, and they are important biodegraders. You would notice that apart from Nocardia, which appears rod-like and actually stains acid fast, these are the two, the Actinomyces and the Streptomyces. They look very much like fungi. They have all these edges, look very much like fungi. And so it's very interesting as to why they are not described as fungi. And so that's a very good question to think about. But if they are described as prokaryotes, then if you think about it in terms of the cellular constitution and all that, that is the reason why they are not classified as fungi, because fungi are eukaryotes, and they are nucleated. So we'll then shift to the proteobacteria. So these are gram-negative bacteria. They are the largest and most diverse groups of bacteria. We have important classes like alpha, beta, gamma, delta, and epsilon. So if we look at, first of all, the proteobacteria, alpha proteobacteria, in terms of the class, many of them would have this structure called prosthecae, the structure that you see here, prosthecae. And it helps them in attachment. It helps them to increase the surface area for nutrient absorption as well. In this class, you have these genera. You have organisms that are able to fix nitrogen. So nitrogen fixers. You have nitrifying bacteria as well, though they are all part of the nitrogen cycle. You have the purple non-sulfur phototrophs. And then you have some pathogenic forms, and then there are other groups of bacteria under this group as well. So if we look at the nitrogen fixers, they will generally grow in association with plant roots, and they can convert nitrogen gas into ammonia. They're able to fix it. You have the genera Azospirillum, which is associated with tropical roots of grasses. So if you look at tropical grass plants, they have roots that you would typically find Azospirillum in there. it will increase the root hair. It's a symbiotic relationship in that they're able to get nutrients from the plants, and then they're able to fix nitrogen for the plants as well. And then you have rhizobium, which is also associated with leguminous roots. So if you look at plants such as soya beans and all the lentils, and they help in stimulating their growth because they fix nitrogen for them in the form of ammonia. So you can see how the roots of plants would appear more, increasing root hairs, and it has a lot to do with, if you can see all the flagella that are attached to these bacteria. And then in this instance, they are attached to the roots of the plant, and so they're benefiting from the food that is available to the plant, and they're helping the plants by fixing nitrogen for them as well. Then you have the nitrifying bacteria, which also belong to the alpha proteobacteria, and these are chemoautotrophs. So they will use inorganic chemicals as a source of their energy, but then they will use carbon as the only source of their carbon is synthesizing their food. What they will do is that they help in the process of nitrification, of converting either ammonia or ammonium into nitrates. And so the plants will be able to take the nitrates and be able to use it. There is a step in the process where there is a primary nitrification process and a secondary nitrification process. So it is first converted to nitrites and then to nitrates, and they play important roles. So an important group among this group of nitrifying bacteria will be the Nitrobacter bacteria, and that is how the Nitrobacter bacteria appears. Okay. Then you have the pathogenic groups. So for the pathogenic genera, you have Rickettsia being a genera, and Brucella being a genera. For Rickettsia, they are aerobic rods, but they are intracellular. They mainly live in cells of mammals, intracellular obligate parasites. So if they are outside the cells, they cannot live by themselves. Important among this group is you have Rickettsia typhi, which causes murine typhus. Murine typhus is transmitted by rat fleas. And so whenever there is an increase in the population of rats in any vicinity, and where you have the causative agents being Rickettsia typhi in the environment, then most likely you see transmission of this particular disease and an increase in its incidence. So this is Rickettsia intracellularly in both pictures, as you can see. So this is Rickettsia, and that's Rickettsia as well. And it lives within the cells of, or within the tissues of mammals. Then you have Brucella, which causes brucellosis. Brucellosis is a highly contagious zoonosis. Zoonosis is a disease that is generally transmitted among animals, but could also be transmitted to humans. And so brucellosis is typically asymptomatic in cattle and the likes, but if you should harvest their milk and you don't pasteurize it, or you undercook the meat from these animals that are infected, then you can also get an infection. Or if you live in close proximity to these animals that are infected, the bacteria may become aerosolized and you may also become infected. And that's how Brucella appears also under preparation. Then you have the beta proteobacteria. These are low nutrient survivors. You have pathogenic groups among them being Nyseria, Bordetella, and Becodaria. Becodaria will appear this way when prepared, as you can see in the picture. They are important organic recyclers, so they are heavily used in recycling, but they are also implicated in the genetic disease cystic fibrosis, where they may cause pneumonia in people who have cystic fibrosis. For Nyseria are Diplococcus. You find them in areas of the body that have mucus. So in the vagina, they are commonly found there as well when individuals have infections. Then Bordetella, which causes Bordetella pertussis, which we have the DTaP vaccine that serves as a protection against it. Nyseria is a diplococcus, so it appears in pairs, and these are presentations of some of the symptoms of Neisseria gonorrhea. Then for Bordetella, they are rods, but they have flagella, as you can see. So this brings us to the end of the third part of our presentation on prokaryotic classification.